This is my video for Ebola virus disease. So just to start off a little bit of the history, EVD was first discovered in 1976 when two outbreaks of hemorrhagic fever turned deadly in two parts of Central Africa. And since 1976, an additional 25 Ebola outbreaks occurred mainly in Central Africa. The first outbreak occurred in the Dominican Republic of Congo near the Ebola River, hence the name of Ebola virus. And the second outbreak occurred in South Sudan. And then from 2014 to 2016, there was an Ebola outbreak in West Africa, which is also the largest one that was ever recorded. And Ebola virus is believed to be animal-borne, with fruit bats and non-primates as the likely source. Once there was kind of two back-to-back infections, that's when scientists sort of started to discover that there were two independent outbreaks, and they really began to study the reservoirs of infections in insects, plants, and animals. And there was actually a very famous book by the author Richard Preston, who it was called Hot Zone, and they basically followed an amateur explorer named Charles Monet, who lived in Kenya, and he went to go explore Kitman Cave near Mount Elgon, and he started having very bad symptoms. He and died of a hemorrhagic attack, and it was kind of the first time that they saw that Ebola was actually infecting humans. So for the taxonomy, the phylum of Ebola virus is Nagarnavericota. Order is Mononegerviridalis. Family is Filoviridae. Genus is Ebola virus, and species is Zaire Ebola virus. Some common names that you will hear it being referred to is Ebola virus or EBD, sometimes also called EBOV, and another virus that has similar symptoms to Ebola virus is called Marburg. So for the characteristics, EVD is an enveloped negative single-stranded RNA virus with a genome encoded for 7 separate structural proteins. EVD contains a nucleocapsid, matrix base, and an envelope. EVD is approximately 80 nanometers in diameter and approximately 800 to 1,000 nanometers long. And something that's kind of unique about this virus is that it kind of creates like a 6 in its genome, and I thought that was kind of interesting. So there are 6 notable strains of Ebola virus, which include Ebola virus, Sudan virus, Typhus virus, Bundibugyo virus, Reston virus, and Bombale virus. Something that is unique about this is that out of the 6 strains, only 4 are known to cause Ebola virus, which is the first 4 I named off. Reston virus is only it is Ebola virus, but it can only be spread from primate to primate. And with Bombale virus, researchers are not sure if it actually causes disease in humans and or animals. So the incidence and prevalence, Ebola virus is most commonly seen in West and Central Africa. And this graph is a little bit hard to see, but this purple outline is basically just the habitat of the fruit bats, which is what the likely source of Ebola virus was from. This light blue is the region in the United States who reported countries that reported Ebola Reston outbreaks. And these little red dots are the Reston outbreaks that occurred in animals. So there's only 3 in the United States. It was very contained in the United States. Actually, there was actually only 11 people in the United States who actually contracted Ebola virus, and they recovered. The deep orange is countries that reported Ebola hemorrhagic fever outbreaks. So you see that's mostly in Central Africa, Ghana, Sierra Leone, and Ghana and Sierra Leone. And the light yellow ones are cases with serological evidence. Serological evidence is basically just they use antibody tests they use blood tests to basically confirm that Ebola was actually in the bloodstream. The little blue dots, or the little blue stars, is the location of reported Ebola outbreaks and isolated cases. So you can see it is still very concentrated in Central Africa. The light blue cases are the cases that were imported from humans. And and that's mainly in South Africa. So who is at risk? So the people who are at risk are mainly just family members and friends who have come in contact with individuals who have been infected with EBD. Also healthcare workers who do not use effective PPE while caring for Ebola patients. So basically healthcare workers, doctors and nurses who went to Central Africa to help or take care of patients who had contracted Ebola. And out of all the research I could do, all I could find was the age that's most commonly at risk, being 16 to 45 years of age and greater than 45 years of age. So for the immune response, so the TIM1 protein acts as a receptor that binds Marburg and Ebola viruses, and the TIM1 protein is expressed in epithelial cells and lines the mucosal surfaces, so the eyes, nose, and mouth. So Ebola virus has the gene GP1 protein, and it encodes two proteins. It encodes a short protein and a long protein. And the longer protein is going to actually be the one that extends out to the viral wall and kind of sticks to the surface. And some of the surface protein actually gets cut off and shed from the contaminated cells, and this shedding depends on TLR4. And the blocking of TLR4 weakens the immune system and actually is what allows the virus to invade and infect the healthy host cells. So virulence mechanisms, EBD is mediated by the spike protein, as I said before, GP1, and it invades type 1 INF responses and actually prevents your adaptive immune response from actually even responding to this virus attacking. And so EBD also has a protein called VP35, which blocks RLR signaling, and it binds to double-stranded RNA. So once these RLR signaling is blocked, your lymphoid tissues are actually destructed, and you lose CD4 plus cells, CD8 plus cells, as well as your natural killer cells. So basically, your adaptive immune system is wiped out by this blockage of the RLR signaling. And specifically in Ebola virus Zaire infections in humans, CD4 and CD8 plus T cells expressing CD95 are actually fatally infected. So for pathology, EBD can remain in areas of the body that are immunologically privileged after acute infection. So immunologically privileged sites basically include sites where the virus is shielded from the survivor's immune system response. Some of these sites include the testes, the interior of the eyes, the placenta, and the central nervous system. In regards to viral attachment, the cells are mediated on the surface of GP1, as I said, and the viral nucleocapsid is released into the cytoplasm, then leading to transcription and translation of the viral genome, and that's how you get these virions replicating and invading other cells. Transmission, so individuals who are initially infected through contact with infected animals such as fruit bats or non-human primates like chimpanzees, monkeys, apes, the virus then spreads through direct contact such as broken skin or mucosal membranes. Like I said before, it breaks that skin barrier, and the mucosal membranes such as the eyes, nose, and mouth is how the infection invades. When people are infected with EBD, they actually will not immediately show symptoms, and an infected person can only transmit Ebola to another person after that person has begun to show symptoms. And it's also important to note that Marburg can only travel from monkeys to humans, while EBD travels from humans to humans. So some ways of transmission are blood or bodily fluids of an infected person. These fluids include things like feces, urine, sweat, even breast milk from a mother, objects contaminated with bodily fluids of a person who is sick or has died. So let's say a patient, let's say a patient is in a hospital bed and they were sick. They leave the hospital bed and another patient goes to lay down and that bed was not sanitized correctly or properly. That bed will act like as one of the objects that were contaminated and can actually get that other person sick. Infected fruit bats or non-human primates. Shared and unsanitary needles were actually a very big problem in Africa. They just were really underdeveloped and did not have the necessary equipment that they needed to treat everybody who had Ebola. And so oftentimes they were sharing needles, and that actually led to a massive increase in infection. Something that I thought was very interesting is also that semen from a man who has recovered from EBD can also work to transmit EBD even after he has recovered and is no longer showing symptoms. So as for symptoms and diagnosis, the incubation period for Ebola virus ranges from 2 to 21 days after exposure. And Ebola is oftentimes hard to treat or is also misdiagnosed due to early signs being really vague. You have things such as headache, fever, and weakness, and they're not really specific to Ebola virus. So a lot of times Ebola virus has a differential diagnosis of yellow fever or malaria or other bacterial diseases. And the symptoms as Ebola virus progresses includes fever, aches and pains, weakness and fatigue, diarrhea, vomiting, and unexplained hemorrhaging. And as far as the diagnosis goes, in order to diagnose a patient with Ebola virus, a patient must have been exposed to Ebola virus at least 21 days prior to the onset of symptoms, and blood samples must be collected in order to confirm the virus. Treatment and vaccines. If Ebola virus is caught really early on, you really don't need as much of an invasive treatment. You can really just intravenously infuse electrolytes into the patient and even just place them on a respiratory ventilator, using medication to support controlled blood pressure, reduce vomiting and diarrhea, and maintaining fever. For blood pressure, just use diuretics. For maintaining fever, you would just use acetaminophen, which is just aspirin or Tylenol, and to reduce the vomiting, you would basically just use like an over-the-counter Pepto-Bismol type of thing. And something that I don't think is talked about enough is psychosocial support. You want to make sure that you have family and friends who are there to support you. And in 2019, the U.S. released the first FDA-approved vaccine called RVSV-ZEBOV, which is given in a single dose and is only protective against the Zaire Ebola virus strain. And in 2020, the ACIP recommended a pre-exposure prophylaxis for individuals exposed to potential occupational risk. Reverse transcriptase chain reaction test is also required, and this test is used to detect trace amounts of EBD in the bloodstream. And note that this test only really works approximately 3 days after the onset of symptoms, because if you really do this any sooner, that your antibodies will not have time to build up anything in the blood. IgG and IgM antibody tests are also used. So prevention, this one's pretty straightforward. Avoid contact with blood and bloody fluids of individuals who are infected. Avoid items that may have been in contact with a person who has been infected. It's also important to avoid funeral or burial practices that involve touching the body of someone who has died of EBD. Also, you want to avoid contact with bats and non-human primates. After traveling to a region that is prevalent with EBD, such as West or Central Africa, it is important to go back home and isolate for at least 21 days. And of course, wash your hands. That's very important. So outbreaks and recent events. So in 2014 to 2016, the first major outbreak was located in West Africa, and it was actually the largest outbreak to date. It had about 26,000 cases, confirmed cases, and about 11,300 deaths. And it started kind of in the rural part of southeastern Guinea and spread across the borders and really just became a global pandemic. And in 2018 to 2020, a new outbreak was actually recorded in the Dominican Republic of Congo, and so much so that the government actually declared a new outbreak with 130 new cases and about 55 deaths, which is a mortality rate of about 42.3%. So for research efforts, as far as the vaccine goes, a 2-dose regimen of a different vaccine that is designed to protect against Zaire Ebola virus is used. Scientists are now testing how long the virus is able to stay in bodily fluids. And for future outlook, the World Health Organization has expressed that they will be implementing additional triage, specimen collections, and isolation measures. The use of antibody therapy for post-exposure patients, and GS5734 being developed for treatment of EBOV. And what this is going to do is kind of allow scientists to see if semen can actually be traced out of the body.